right, welcome everyone. My name is Lila. This is Taxonomy Tuesday for the New Jersey Mycological Association. We meet every Tuesday evening to share photographs of our recent finds, typically. So we do that by sharing from our desktop the photos, or if you have trouble sharing from your device, you can email them to us, and the host, in this case me tonight, will bring them up and we will discuss them. And if you don't know what they are, that's okay. We'll try to help identify them. We don't always know what they are either. So there's a lot of mushrooms out there. So with that, we can get started. If you have some that you'd like to share, you can put your name in the chat. You can find the chat typically across the bottom of your screen here in Zoom, and we'll look at those, and we'll look at the ones that people have emailed in. Does anybody have anything? Actually, if you've done this before, oh, Lauren says she's got some, but doesn't want to go first. All right, Christopher's got some. All right, let's start with Christopher because he's done it before, and the newer people will see kind of how this works. So Chris, you want to take it away?
Yeah, let me just open this thing up. All right, I got a little weird one here. I was out fishing with my dad, and these things are right. I mean, this is about 2 feet from the literal water's edge of a lake. Just growing out of the sand is like all pine trees, and the oaks aren't anywhere close. They're another 40 foot up a hill. That one's got, a real little bit of material attaching there for this one. You could see where it was left after it opened.
Sort of a ring zone kind of left there.
Yeah, I haven't gotten a chance to do anything more with it. I have one sitting, but I mean, if I didn't know better, I would say that it's one of these Lacrimarias. It didn't have like a super distinct gill edge color difference, but it is there.
Mm-hmm.
How big are these? How big are
Pretty small.
Yeah, it's
Right.
probably
Nos.
something in the Inocybaceae. Either Inocybe or maybe Mallocybe.
Yeah, that's what I would have said. Sorry, I'm late. But looks like an inocybe type thing to me.
Yeah, if you smash mount some gill, you should be able to find some interesting things. Lacrimaria spores are really very distinctive.
Yeah, I just showed one not long ago.
Yeah. Also, the spore print would be black for Lacrimaria, but you might not get a print of any of these. So
it left a tiny smudge of brown, like not anything I could even
smudge
take.
of brown. That sounds like an Inocybe. But maybe not a true Inocybe, maybe like Mallocybe. You need to smash mount some gill and see if there's Poroscystidia.
Yeah, I held on to it.
Yeah. Inocybes are nice. They cooperate well with scoping the gills via smash mount because everything is pretty big. The cystidia are big. The spores are not real small usually. So You can probably make some headway with your scope on this. So there is an inocybe I've been finding the last few years, but I haven't found it this year, that has this really prominent coordinate partial veil like you see on these. If you can get to one of those photos where, yeah, see the one on the right? Yeah, look, see, it's got a really prominent coordinate partial veil. I'm going to look up that name now. I'll be back. I don't think it's the same though, because I think that other one was kind of yellow and this one's more kind of brown. So let me see if I can find that name.
Dave, before you head out to find that name, was it
Yeah.
also growing in this kind of ecology? This looks, I mean, this is in sand, yeah.
It's
Yeah,
this place is a
inocybe's
sand
can show up
quarry.
just about any place.
Mm-hmm. But I think that would be an odd place to find a Maria.
Oh, they
That's what I
show up
thought.
any place also. They show up in weird places, like just alongside buildings.
I just the sand, like, I mean, this is a weird environment. I mean, like 1 foot from this is what, this other, like this is the habitat that I found this thing in. This is about 1 foot away from that thing. So like, it's just sand with nothing in it.
Right.
Most inocybaceae are thought to be mycorrhizal. Now that might only be what is believed. I don't know how much evidence there is to back up that claim, but they could be mycorrhizal with plants other than trees. Also, that's a possibility.
Yeah, Chris, do smash some gill material.
Yeah, I saved it because it was too interesting. I just haven't gotten around to it yet.
But yeah, Inocybaceae is pretty much a lock there.
I think so. Yeah, did you smell it?
It wouldn't have mattered because I was like, for once, catching fish. So, it probably just would have like smelled like that.
Smelled like fish.
Yeah. Like I picked this thing up and threw it in my tackle box. So.
All right, well, good. Thanks, Chris.
Thank you.
All right.
So one more genus. Oh geez, I think it's Pseudosperma is the one that Calamastrata used to be in, or is in now. It used to be Inocybe Calamastrata. So that's another one to check. I think it's Pseudosperma. Anyway, I'm sorry.
All right. Are you going off to look for a name, or are you done with that?
Yeah, I'm going to go look for a name.
All right. You can let us know.
At
what
least
that
something
is.
that compares favorably, morphologically. I'll be back.
All right. All right. While you're doing that, Lauren, are you ready to go?
We're going to give this a shot. This is a new computer, so it might fail miserably.
All right. We're patient.
Igor, are you there?
Oh yeah, I'm here.
All right, this is one I think you taught me.
I think we're not getting that one first.
Oh, never mind. Wrong one. But I did also find this.
Can you see this?
Not yet. You need to share.
Sorry.
I found the name I was looking for, Inocybe subocrasia. But I don't think it's the same thing as what Chris found. There are just a few similarities. I'm sorry. Go ahead.
Does this show up?
Yeah.
Okay, cool.
I don't know. I don't see this too often, but I'm wondering if this is still I don't know how you pronounce it. Is it alboater or alboater?
Tylopilus, yeah, alboater.
Is that how you say it? Alboater?
Either way is probably fine.
Looks like it from here.
It's just fun to see. I just saw one. It was pretty.
Yeah, sure looks like it. Did you cut through it?
No, I didn't think I had to because are there any lookalikes?
Well, there's that atronicotanus thing, but that's not as dark as this. And then there's this thing, Grisio virida Grisia, something like that. It's more of a southern thing, but it's also a coastal thing. But I think
Okay.
that one has a reticulate stipe, and I don't think it's as dark as this. So this is probably alboater.
Okay.
Could you please zoom in on this one?
Let's try.
Yeah, I think this is alboater. It looks like there could be some wrinkling on the stipe. I don't think this is reticulation because the lighter colored right-hand side closer to us is devoid of any wrinkling or reticulation, and the cap is velvety chocolate brown. So yes, I think this is it. Grisiocarnius, as Dave said, is a dweller of the pine barrens, likes the pine, I think. And it's just overall sort of blackish, charcoaly kind of a mushroom with a gray pore surface, and it is reticulate, unlike alboater, which might be reticulate just at the very apex, which is common for a lot of boletes. It's not a reliable field character anyway. So yeah, I think this idea is almost certainly alboater, unless we have a lookalike that we haven't come across by sequencing yet. And this is not an easy genus to sequence, by the way, especially by nanopore. So that's why we're not seeing a lot of data for Tylopilus in GenBank.
Okay.
If you handle this one a lot, it will stain your fingers black.
Oh. That's cool.
That's
That's
probably
probably
The
one of
inner-
the best edibles in the genus, together with Badiceps.
This is one of the best edibles?
In the Tylopilus genus, yeah, but this
Oh.
is I mean, I don't think it's on par with Edulus, but it's a very, very good edible.
Oh, okay, good to know.
It-
I guess
it
if I did know
tends-
that at some point, I forgot.
It tends to be very firm fleshed, so it's good to use fresh. Whereas some of
Okay.
the porcini mushrooms, once they get a little blown out, they're very good after they're dried and rehydrated, but fresh, they're kind of flimsy. So this one, if you find nice, fresh, firm material, you can slice it thinly and put it on a grill even.
Okay.
Hmm.
Good to know.
yeah.
And
But
it's not bitter.
Igor, I miss.
Oh no, it's great.
Wow.
Wow.
I enjoy these a lot.
Huh.
I
Lila, how often do you
misstated.
find them?
I used to find them quite a bit, but I haven't seen too many in the last year or two in my spot.
Hmm.
Yeah.
Did the sun color the stipe like that?
It could be just oxidation because they do stain blackish when you touch them, and when you slice them, the flesh also turns dark brown. Grisiocarnius, I think, stains red before it then fades to blackish on the
Yep.
flesh.
Igor, I think this one stains pink also at first.
Yeah, you know what? I remembered. I wasn't sure, but yeah, eventually that pinkish color turns brown or blackish brown. But yeah,
Yes, it changes from pink to a darker color in not that much time. And that Grisiocarnius, I just wanted to mention, if you want to eat these, you should at least familiarize yourself with Grisiocarnius if you're in an area where it's known to be found, because that one has a bad reputation.
well,
Oh.
they don't overlap. You don't find bodies alboater in the pine barrens, and there are plenty of nice observations on the Mo of well identified Grisiocarnius. It's a very striking bolete. You are not gonna confuse it with anything else in the genus, and besides, it's not even in Tylopilus probably anyway. I tried to sequence it many times. It just doesn't produce anything.
Hmm.
So, but yeah, I have sequences of alboater, and it's Tylopilus is a very diverse genus, and ITS is a wonderful barcode for these things because there's just absolutely no overlap. I mean, everything is very unique, and when you build trees, everything just falls into a multitude of these mini clades inside the tree. It's just amazing how this genus is structured globally in terms of what the relationships are between species and how they clade with regard to each other in the tree. So yeah, it's a poorly understood, fairly large genus that is yet to be unraveled completely. So we're just looking forward to more results.
Cool.
So next time you find it, I suggest you do cook it, or dry it. It's a wonderful edible.
Did
Oh,
you
cool.
get a gill shot or a pour shot? I mean,
No.
No, because I was pretty sure that's what it was, and I just didn't want to mess with it. I figured you guys would all know
yeah. Okay.
what it was. No, I didn't feel like messing with it.
All right. The pores, for those of you not familiar with it, are pale. It's almost white
They're
underneath.
whitish, yeah, and
Yeah.
then they turn pink in age.
Yeah.
And they also bruise brown, I think. So
Yeah. Yeah.
thing is,
Okay,
if you do
good.
microscopy on that one, if the pleurocystidia have kind of a broken glass appearance inside that stains brownish and KOH, most certainly it's a feature of Tylopilus across the board. So, like I said, it's a very diverse genus. So even if the DNA sort of puts it like in a separate corner, and you cannot find you might think it's a different genus altogether based on the topology of the tree. Most likely it is a Tylopilus, but Grisia carneus I'm not so certain about. Anyway, let's move on.
Okay. All right. You taught me this one in person. I think I'm gonna get this right, but obviously you let me know if I'm wrong. I think this is Boletus vermiculosoides.
That looks like from the top, that's what I thought, but we have to see. Yep, I think it is. And that is going to be a Boletus. I mean, I've known this already for years after sequencing these things, but the type collection was finally sequenced, has been sequenced some time ago, and it is claded in Boletus. So it's going to be Boletus vermiculosoides.
Okay.
Yeah.
And you taught me to cut this in half and look at all the worms.
Yeah, vermiculosoides suggests that it is, infested with vermin or these critters or larvae. But there's also vermiculosoides, and we don't know what that is because it's a name by Peck, and nobody sequenced that ancient holotype. So
Okay.
we know genetically what vermiculosoides is now, but we don't know whether that's the same thing as vermiculosis. And again, this is just a matter of time before somebody decides to invest time and money into getting it done.
Which was described first, Igor?
Oh, vermiculosis, of course. That's 19th century. Vermiculosoides is a Smith species from Michigan, dates back to the 60s, I think.
Okay. And they're American species.
Absolutely. Yeah.
Okay.
There's absolutely no overlap in Boletus with China. We don't share anything in America with anything else from anywhere else in the world from that genus. Everything in this continent is unique for Boletus.
How would you describe this smell? Because I did smell it and it smelled good. It smelled like food.
Many of these boletes do smell pretty good. So I think subgraviolens, which might look similar to this one, smell like boiled urine, according to Smith. I'm not sure how he came up with the name. Why would you boil urine? Doesn't make any sense. But yeah, I'm not surprised that this smells good. They all do, for the most part,
Yeah.
with a few exceptions, of course. Yeah, but the reddening of the stipe at the base in the exposed context is very common in these species.
All right. Well, that was it. Thank you, everybody.
Yeah, rosewood oak, broadleaf trees.
Yeah,
Upland
there were
forest.
oak and it was right in front of a beech tree, but it was in a spot with oak and beech and birch.
Mhm.
Right.
I just want to say that boiling urine used to be a source for an ammonia-like substance for fixing dyes or changing the color in dyes,
Oh, the urea problem,
mushrooms
right?
or other things. So it's not unheard of, particularly with lichens too.
Well, now I understand where that description probably came from.
Always kind of wondered why it was in there. So
Yeah.
thank you. Wondered why people knew what that smelled like.
Yeah, but it's still curious why Smith would know that.
Making
Peters.
making saltpeter for ammunition.
Yeah, that's what I was gonna say too, right?
Yep.
Yeah.
I did not know that.
You
Yeah.
have to boil a lot of urine to make gunpowder.
Honestly, it's
Well,
not nearly as much as you might think.
there's references in Karen Castleman's lichen stuff of piddle parties in the late 1700s of get the men really good and drunk and get them all to piddle so there was enough urine collected for whatever they wanted to use for dye. And maybe saltpeter, I would suspect.
Cool. I love how mycology and history and culture intersect. And we get tidbits of it all the time. Thank you. All right, let's move on. I think Nancy and Karen, I think you were next in the chat. Would you like to go?
Sure, happy to. Let me see if I can make sure these are pulled up here. Share photos. All right, so this was one of them. Huh. If I don't know whether how to do this. I'm on an iPad. Oh, there we go.
There you go. There you go.
All right. That's the bottom of it. Let me just see whether I can. Huh. Why is it not?
Even just from that picture, it looks like it might be Tapinella atrotomentosa. I'm guessing by the fuzzy black stem.
All right, I'm going to try this again. Can you see my screen now or not?
We see lots of thumbnails of photos.
Yeah. Okay. That's
Nice.
okay. That's the one. Yeah. So that was the.
Okay. Looks like the
This was the
top.
bottom of it, and that's the top of it. Yeah. So
Let's
Looks
see
like
the
it's on
bottom.
a stump also. Is that a pine stump?
it is on. Yeah, it's on a stump, and there's a lot of different. Like there's a whole bunch more on the stump. I don't think I got those. So.
I thought I saw a picture that showed more of the stem. Maybe
I
not.
don't.
I think it was the one that we were just on.
I think
The first one. The first one that
this
you
is the
showed.
same thing. This is on the same. Is this the same stump or not? I'm sorry. Yeah, no, it was this one for sure.
No, and then
the bottom. And then that bottom.
It looks like neolentinus a little bit too, if that's not a fuzzy stem.
Neolentanus has scales on the cap.
I see what Susan's saying too.
Susan, you're right. That's what this is.
Yeah, I agree.
Yeah.
What do you think it is?
Tapinella atrotomentosa. My interest in that, of course, is because it makes a wonderful dark green dye for wool with iron as the mordant. Thank you, Sue. for putting up the name in the chat.
Oh, thank you. Yeah. And then we did find this, but I actually put it on Facebook, and someone told us it was a crown coral, and we ate it tonight. That was good. But then there was this one also. There's a couple of others like this one. We see these a lot, and we're just sort of curious as to what they are. That's the bottom of it. That's the top.
Potentially Trametes and potentially Trametes lactinea. But we need a better shot of those pores.
I was just gonna comment on the angle of your shot. You want to see the shape of the pores.
Okay.
So, like, as you're
I don't think
photographing it, like as you turn the mushroom a little bit, you'll be able to all of a sudden you'll be looking at all the pores. That's the photo you really want.
Okay. Okay. All right.
This
Yeah,
is
we're
all
really new
good
to
so
this,
far,
so-
though.
Yeah, and when depending on how new you are, when I say pores, I mean the underside of the thing.
Okay.
So
Because
when you're
that's
look-
this is the underside of it.
So can you zoom in like to that edge right there?
I don't know.
Yeah, and it's hard to tell from that photo whether the pores are circular or angled.
Oh, okay.
You know.
So that was a question that we had, and and then this was the last one. These guys were the last one that we were curious about. That's the top, and then that's the bottom.
They had long, skinny, like semicircle stems coming out of the
Pluteus.
trunk.
I don't know.
Pluteus.
Are those gills free or are they attached?
Oh, that's a good question.
Look,
Because this could be Melanoleuca.
They do look
Although the gills
attached,
are a
Dave.
little bit wide for Melanoleuca.
Nancy, can you zoom in on that point of attachment of the gills and the stipe? Yeah.
Like right there.
Oh yeah,
Oh, they're free.
they look away.
Oh, those are attached.
What?
Oh, they're attached.
I think
They're not broadly attached, but they're attached. Pluteus gills are rounded back on themselves. You
yeah,
know,
I think it's a
like
trick
a
at the end.
millimeter or two from the stipe.
I think
These
it's
are
a
tapered and
trick.
okay, the stipe is a little bit flared, so it looks kind of like it's not attached to the. But you see where those are attached. I don't know what this is, but I don't think
A lot
it's
of them.
Pluteus.
Yeah,
And we were hoping that they were edible, but we're not eating anything we don't know anything about.
yeah, that's
It could be Melanoleuca.
I think this is probably Rodmanii.
The gills don't
Megacolibia
look quite
rodmanii.
right for Rodmanii. Rodmanii has those thin, wide gills.
Well, this is
I
a
don't-
close-up, so there's some distortion.
Yeah, I don't think this is Megacolibia. I think this might be Melanoleuca. Some kind of Melanoleuca.
Yeah, I don't think it's the Megacolibia at all. The gills are not far enough apart or deep enough with little flesh. Yeah, that's not, that I've been looking at them today and they don't look like that.
Okay.
Those don't look like Megacolibia gills. They look more like Melanoleuca gills to me. So Melanoleuca really, it's got a white spore print, so that's not going to separate it from this other thing we're discussing, Megacolibia rodmanii, that also has a white spore print. Probably need a microscope to really, solidly find out whether or not it's Melanoleuca.
Well, the gills are shaded pink, and they might be free. So another possibility that this is a Pluteus of some kind.
It's not a Pluteus. It's not a Pluteus.
Was that little
Those
one?
gills are attached.
We're thinking outside of the box. I'm not sure this is Melanoleuca. I don't think it is.
What?
The gills are much closer for Melanoleuca than this.
Yeah, the gills are, for most Melanoleuca species, the gills are really close. These are pretty mature, and there are, there's more than one Melanoleuca species that's found around here, but that's true. The gills are white. I said that right from the start. The gills are a little bit widely spaced for Melanoleuca.
And they're pink. And there was also a photograph shown for just a brief couple of seconds of a young mushroom that looks more like a Pluteus to me.
That's what I wanted
From the
to
same
know.
log.
Was that little one on the same log?
Or there might be a different mushroom growing with them, because I don't think those are Pluteus. Those
Go
gills
back.
are attached.
Yeah. I don't know. They kind of looked free to me, those gills, but it's hard to be certain. So Nancy and Karen, don't be afraid to pick these and turn them over and get them in some nice, soft, indirect light and focus in on the gill attachments.
Now that looks like Pluteus.
That looks like Pluteus.
Yeah.
So,
but that's not the same as those other
it's not. This is not the same. It's a different one.
Same log?
Yes. This
maybe.
Yes, this would have been the same log.
And this is the underside of that one. We're
Is
just
it
looking.
possible to zoom in on the gill attachment here?
Yeah.
It's. But do you see how it looks like a little annulus that's smooth in between where the gills double back on themselves and the stipe? There's actually a little smooth annular region in between. The gills are not, they don't taper and sort of peter out, on their way to the stalk. They double back on themselves, and there's always, especially with a mature Pluteus, you'll see this little annular region that's smooth.
So the question we had was, any of these edible? But obviously, if we don't know what they are, then we're not going to eat them. So.
Well, the Pluteus is probably edible. Pluteus section Pluteus they're typically called deer mushrooms, and they're popular edibles over lots of different cultures.
Okay.
But you have to make sure you positively ID, because there's a few things that could easily be mistaken for Pluteus for a variety of different reasons.
So when we had a question, because we're hesitant to pick things when we're just walking, because we don't want to disturb nature any more than we need to, right? So it's just like when we're walking and we see something, but you said in order to really ID it, you need to pick one and take pictures of it and really, is that the sort of
go for it?
Is that what we're supposed to be doing?
Yeah.
You have to really look at it. You have to see the underside. You have to see the base of the stem, the attachment. Notice what kind of substrate, whether it's wood, whether it's in the ground. You have to look at a lot of little details. Also, Igor mentioned the velvety surface of that bolete. So, whether it's smooth, whether it's rough, whether it's scales or what. So the best thing is to really pick it, turn it over and look at it. Okay. And then if you can, take the time to make a spore print, which often means taking it home. But still, if you're really going to get good at noticing all the little features, you have to really look at it.
Okay. All right, we'll start.
Picking a mushroom is analogous to picking an apple off a tree. You're not really damaging the organism that produces the mushroom.
Yep.
Yeah, that's a good point.
I was going to make the exact same analogy.
So they'll just grow back the next year, essentially.
Oh, maybe not. Those Pluteus are saprophytes. They're living off of decaying wood, and once the nutrients are used up in that wood, that species probably won't fruit on that wood anymore.
Okay.
Now, some mushrooms do occur annually, given the right weather conditions, and the more or less the same time of year, because there are some mushrooms that come as the fruit as a result of a fungus that associates with a living tree in a long-standing symbiotic relationship. So there's different types of ecology associated with different types of fungi. Basically, 3 types of ecology: saprophytes, parasites, and mycorrhizals with some sort of species possibly playing more than one role.
Okay. Great. Well, thank you so much. I appreciate this.
Yeah.
Yeah,
Great.
and one more point on that, because I appreciate your, hesitation to disturb the natural environment there. My rule essentially is if I see a couple of them and at least one of them is in maturity, so for that potential Pluteus Melanoleuca that we were just talking about, if that cap is completely open and it's a mature mushroom and there's a couple of them around, you're not doing any harm to that organism by picking a mature one. So.
Yeah, I think the only counterexample I would suggest is a perennial polypore.
Yeah, yep,
that's
sure.
growing on a tree and the lifespan is years. So harvesting something like that is having a detrimental effect on the what the purpose of that thing is. So I usually try to leave perennial polypores alone.
But Dave, like a capitate, stipitate, a mushroom, capitate, stipitate, guild thing growing on wood or on the ground, if there's a bunch of them growing and some of them are mature, I don't think you're doing any harm to that population by picking a few.
I don't think so, and that's the prevailing attitude amongst mycologists. But over harvesting, in particular of edible species, is sort of a question I think that nobody really quite understands how to address, to be honest.
All right. Before we go down that rabbit hole, let's keep looking at mushrooms here. Shihong, you said you have at least one?
All right, let me share.
Okay, and you're very soft.
Okay, it is better?
Yes.
All right. Oh, I don't have my mouse, so can you see my screen?
Yes.
All right.
My naturalist.
Yeah, this is what I think it's interesting. It's the first time I saw this one.
Right. Can you make it bigger? Can you enlarge?
Let's see. It is better?
Yeah.
So last week I was at Nova Scotia, this at the Kijimocook National Park. I was surprised. It was a lot of pine tree with river and the lake, lots of moss. But I was surprised I did not see many mushroom. This is one of the 5 mushroom I saw. They're only a single one, so I took this picture. I did not pay too much attention. I thought it's some kind of a reishi, but I put it in iNet. It gave me a totally different name. Yeah, so I was totally surprised. It's a Turbinellus floccosus.
Correct.
Yeah.
I agree.
AI
Yeah,
AI is correct for a change,
Yeah. So
but that's an easy one.
so this is the first time I saw this one. What did it look like when it's mature?
Well, it opens out a little bit more of a vase shape. This one's still kind of closed in a little bit at the top, but it will open up to be more flared at the top and sort of almost like a funnel. And the scales inside will be the same color as the very top circle there, kind of orange and scaly. But this is a young one, very nice looking.
Yeah, so this is I'm very glad I saw this for the first time. I was just hoping to see something mature. Is it edible or what can you use that for? Is there medicinal use?
Some
Oh,
people eat
okay.
these and some people get sick on them.
Okay.
Yeah, a lot of people confuse them for chanterelles, right?
Yes,
Yeah, they're called like a false chanterelle sometimes.
yeah. If
Right.
you've seen it next to a true chanterelle, you would not mistake it, but
Right.
until you get familiar with it, you might mistake this for chanterelle.
I think
I
they call it the woolly chanterelle as well.
I think they call scaly chanterelle. Yeah, scaly
Scaly
chanterelle.
chanterelle. Yeah,
Yeah.
the fertile surface, the veins will spread out a little more when this fruit body spreads out and becomes mature. And then the outer surface is reminiscent of a chanterelle. That's probably why those mistakes are made.
I see.
They can
Yeah,
get big.
I don't
They can get
find them.
big.
They can get big, yeah. I don't find them so much really in Jersey, but I do find them in the more northerly latitudes.
Same. I don't find them where I live in the oak woodlands, but I've found them in more mixed woods in Wisconsin and further north.
Mm-hmm.
Mixed words. Okay. So
Yeah, I've already seen these this year. I think like 2-3 weeks ago. In mixed woods, beech, hemlock, maple, birch.
okay. So I think the woods I saw have mostly pine, some birch. So that's counted as mixed woods, yes.
Mm-hmm.
I found them in with mostly conifers as well, hemlock areas.
Yeah, I can share another one. Not too excited, but let me see if. Oh, this is a good looking bolete. I think I know what was the name before, but I just cannot remember.
I just found this today, if it's what I think it is.
Yeah.
Hemilexinum subglabripes?
No, no, it's not.
No,
No.
looks a lot like,
This is going to be Pulchroboletus. What is it? It's a frost species. Dave, what's that thing that you collect with hemlock in your area?
Shihan, can you zoom
Oh,
in?
geez, I thought it might be subglabripes, honestly.
No, it's that thing that stains blue that grows with hemlock.
It
It's a
does,
northern
yeah,
species.
it does Bruce blue, yeah.
Oh, Dave, come on, you know that species. What
Ugh.
the hell is its name?
Oh, it's a miniato-olivaceus.
Yeah, miniato-olivaceus, yes.
Yeah, Boletus miniato-olivaceus. Okay, I can go for that. That looks right.
Now, is
Yes, that
this
looks
like
right.
North Carolina in the Appalachia in the mountains?
No, this is Nova Scotia, Canada.
Oh, that is Nova Scotia. Yeah, this is then miniato-olivaceus.
Yeah.
Yeah, okay, I can go for that. This looks more like that species than subglabripes.
Can you type in
Subglabripes
the
has a different
species
kind of
name, please?
stalk.
Yeah, I guess AI did not give you that name, did it?
Yeah,
It did not.
I was going to go put it in the chat, but someone say it again. What is it? Miniato-olivaceus?
Miniato-olivaceus. Okay, I already put it in.
Nice.
Yeah, and it's going to be Pulchroboletus, I think, based on what I know.
Oh, that's what threw me off because I forgot about this generic transfer that's underway. Yeah, that did not ring a bell with me when you said Pulchroboletus.
Yeah, the new name, I'm going to type it in. Will be.
Are they going to retain the species name?
Absolutely.
Oh, that's good.
can we see the top again?
Okay. Let's see. Okay, that's not top here. Yeah.
Yeah, okay. Yeah, okay.
Oh yeah, absolutely. See the little like olive stains, olivaceous stains here and there on the like just areas that are kind of greenish with sort of a pinkish reddish ground color.
Okay.
Yep, absolutely. Yeah, good call, Igor. That's what that is.
Thanks. I just got a mental block with that name. I mean, I know the species very well, and eventually it would come to me, but I don't see this thing very often. So
Yeah, you just threw me off a little with that Pulchroboletus because I had forgotten about
Well, I mean, I've done the
Boletus?
sequencing, and that's my reference point. I mean, I know what I
Okay,
sequenced,
yeah,
and I just go for whatever the new combination is most likely.
yep.
Yeah, so
So currently, this will be found in field guides under genus Boletus, which is sort of a joke, really. I mean, this is not anything like the actual Boletus species in the modern sense. 100 years ago, maybe.
correct. Yeah, everything was bunched into Boletus, 50 years ago. I mean, that genus is now, in a strict sense, is reserved for porcini, like Edulis and anything related to Edulis.
This one has a bad reputation as it being a sickener.
Okay.
Yeah, Frost described it, or actually was Peck who described it. It was Frost's collection. So when it was described by Peck, Frost was the name, the author of that species. It was described, I think, from Vermont. And Professor Halling actually visited the type, the area, the geographic area, the location, the locus typicus, where the type was collected, and he gave a very precise modern description of that species, which was pretty much in line with what Peck stated in a very succinct way. So we pretty much sure about the identity of Frost's bolete. This is the only one that looks like it. There's just nothing closer on this coast, that is.
All right. So I will stop share.
Nova Scotia, that's pretty nice.
Yeah, it's a beautiful place.
Super. All right. Thanks, Shihong. Brandon, you're next. Are you ready to go?
Yeah, I can go.
And after Brandon, Paul, you're next, and then we'll go to the email.
Can you see my screen?
Yes.
Well, I guess we'll start with this one that I found today. Slime mold. I'm calling it Physarales. Maybe it's album, perhaps, but I could be wrong. This is under the stereoscope. You can see the cracking, kind of the capillation. Not quite out yet. This one is a little more immature, hasn't started cracking. And then, on a foot. Even for a slime mold, these were tiny. I found a bunch of slime mold on this one log today. And this was easily the smallest one. It was probably 1.5 millimeters tall. And you can see it's taller than it is wide. I don't know if any of these kind of caps were more than 1 millimeter wide. And I'm also not sure if this is actually flat, because I saw some that were flat and I saw some that were more kind of globose on the top of that foot there. This one in particular, this little tiny piece of wood rolled over, so I'm not sure if that got compressed or if that is indeed kind of a flat head on top of the foot. This is a bad photo, but you can almost make out the capillation coming out there. So I should be able to get some mature spores out of that one there. And this is the in situ photo taken without a microscope, or a stereoscope. Really can't make much out, but they're real small.
On collection of this item, it looks like they're in pretty good condition by the time you got them home to your stereoscope.
Yeah. I mean, I only was in the woods for like an hour. I only got 50 feet from my car. So I just kept putting things that I found directly in my car without having to carry them around all day or for a couple hours or something like that. So they remained in pretty good condition.
Nice.
But yeah, Virginia, what do you think?
I don't like to make suggestions without looking at them under a microscope to look at the capillicium and the spores and more or less, and to see whether it's the outside peridium, whether it's solid or more sugary crystalline. So I don't like to make a prediction on just photos like that.
I mean, could it really, and like I said, I found it today, I'll do all that, but could it be anything other than Physarales? Like it's either
No, I don't.
it's either Physarum or
Fissarum or
the one that starts with a D, right?
didymium if it's crystalline, if the surface coating is more crystalline, it could be didymium, it could be diderma. And that all depends on what the capillicium looks like and what the nodes look like in the capillicium.
But
So
all of that is in Physarales, right?
they would, yeah, I think they're all in Physarales.
All right. So I'll do a little more work on it, but
Yeah, so
at least I'm in the right order. I know it's, getting something to an
Yeah.
order.
yeah, dark spores, generally with a limey surface, you're pretty safe in Physarales.
All right, good. But yeah, I do. What else would I be looking for to determine between what is it? It's not didymium. What was the one that starts with a D?
Oh, there's didymium, which the surface lime would be crystalline, which it looks like little stars.
Right. Yep.
If it's Physarum or diderma, there probably would just be amorphous, not much shape to them.
All right. Cool.
The crystalline lime is a giveaway for didymium.
Right. I remember you.
I
know about finding the lime under the microscope, so that's a good way to separate the two. Appreciate that.
I have one of those, and I brought up the picture. We could look at it later of the crystals you're looking for.
The crystals would be in the lime, and they're pointy, star-like.
Yeah.
Yeah, they look
Sometimes
like,
you'll see one where the coating is almost looks like it's covered with sugar, and that's quite likely a didymium, because that's the way the coating would look on it, not as solid as in Physarum or diderma.
Nice. And I know you don't like to put a name to anything. That's why I just
I don't put a name to
anything. I just put it to Trichales on this one. It might be an Arcyria.
Let's see close. It's more like the monotales, or it could be they're not clustered enough to be stemonitis.
That's what I was thinking.
They could be Arcyria, which I don't recognize it by the color.
Yeah, I thought that was weird.
It's not a usual, Arcyria cinerea is whitish or grayish. Then there's pink ones and red ones. And that could be one that I don't know right offhand.
I will say it did have a little bit of a brick red quality to it. I thought this might be young growth here. This is an in situ
I see
photo.
the pink ones in there.
Yeah.
They could be an Arcyria. They would not be Camarops or anything like that.
But let's go back to this photo where you can see the foot pretty well. I don't know if these things are joined. I mean, maybe they are, but but yeah, I mean, we got full capillicium just in that shape, that
Yeah.
cylindrical shape, you know.
Uh-huh.
Um,
And I would do microscope work and look at the spores.
I will. But again, I
And
found it today, and I'm
if it
glad.
has
I'm glad you're here
surface
because
net. Uh-huh.
one thing that I did want to ask, this you can see it in the macro, right? All of them have what I thought in situ was maybe a little hole at the top, but it
They're
seems
not whole. I don't know. It's a
yeah.
dark. It's like maybe didn't quite develop or dry out. It could have been.
Or like it's a little bit of that peridium. Am I using that correct? That got stuck at the very apex.
I don't know if it usually does that. No.
Hmm.
It could be just didn't dry out right.
Yeah. But again, found it today. Pretty cool, I think. When comparing to Arcyria, one of the things that I was kind of looking at before doing any micro or anything was because I found what I'm pretty sure is Arcyria cinerea today as well.
Right.
And the the capillicium.
Our Syrias sometimes puff out like they're expanding a lot. And Comatrica, I don't think does that. Or
okay.
yeah.
Okay, nice.
Mm-hmm.
Well, collected. I'm hoping this one seems to be.
If it's so, it would not be Trichales. If it's a Comatrica, it wouldn't be Trichales. It would be Demonitales or something. I know that's the old one. There were 5 different orders, and now they made a bunch of new ones, and I can't remember what the ones.
Okay. Yeah, because I thought I was being safe just calling it Trichales. Like, it's got to be one of them. But yeah,
yeah,
I got to look into that.
Yeah, if it's Arcyria, it would be Trichales, unless they change that too.
What a great find.
Thank you.
Okay, so I see your white one isn't quite white. It looks grayish.
So this is under the scope, which washes out color.
It doesn't.
Let's go to the in situ photo, which tells you almost nothing.
Oh, that looks, yeah.
Other than what the color is, right?
Whiter. That looks whiter, like
Yeah.
it might be Arcyria cinerea.
So yeah, this is the in situ photo, which
yeah, I would say that looks like Arcyria cinerea.
And then when you go under the scope, it washes it out a little bit, makes it look a little darker.
Yeah, sometimes it changes colors.
Yeah, but you can at least see, longer. The foot is almost equal to the exposed capillicium. And yeah, this photo looks a lot darker, but I can assure you.
Yeah, that makes it look different. That's hard to.
But I can assure you it's the same thing. So I feel good about the ID. I mean, I collected the thing. What do you think, Virginia?
If those brownish ones are all from that same collection, then I guess they have to be Arcyria cinerea.
Right.
But that's what they look like right there.
Yeah, this might be a better example.
Yeah, that's a good picture that shows them pretty well.
All right, cool. Hey, we got one without doing the micro. All right.
Well, you should do it anyway and learn to see what the capillicium looks like.
I will. I will. And then one that I have done the micro on, my weekly Orbilia, that I put a name to a thing this time. I know it just says genus Orbilia up here in the what I've assigned it, because they don't have the name on iNet, which is, I keep wanting to say deliculata, but it's not. It's delicatula. Sorry, delicatula right here. So this was found on a branch about 2 centimeters wide. It was partially submerged in a muddy bank. This is different from the one that I shared last week, by the way. You can see this one's got a bit more of an orangey translucent quality to it, and it is much, much smaller. So this is the biggest one here. I would say that's about 1.5 millimeters, maybe up to 2 millimeters. But the majority of them were barely 1 millimeter. Some of them were less than that. I had a bit more luck, and you might have to bear with me on this because I really had to. The micro is really tough, but this is the ascus that we're looking at. And if any of the new people have stuck around long enough for me to work through my thoughts, an ascus is kind of a reproductive structure that you look at under the microscope that can have unique qualities, and within them they contain the spores, which even more variable across different families and genera and stuff. But here you can see this one's curved, this one's angled, and it's out of focus, of course. The next photo shows the exact same thing, just trying to show different details. This is the same ascus as this one here. And you're probably thinking, Brandon, what are you talking about? I don't see what you're talking about. But when you look at this micro-Quebec observation of the thing, that looks pretty familiar to me. That looks pretty familiar to the thing that I was just showing. And then above all, look at this. This is what the asci look like, with some immature spores within them. And they're all over the place. They're angled, they're bent over backwards. Here they are outside of the ascus. And all the measurements fit for the name that I used, even down to the petersii and the asci being, I think I had 32.5 as my measurements. And those spores are, they're well below 5 microns, which I don't do well measuring that small because I only do it at 430x. Sorry, I lost it there. But yeah, put a name to an Orbilia.
So great.
Yeah, kudos for working on that tiny stuff.
Yeah, I mean,
Easy.
this is at 450, so that's an individual spore. It's immature, obviously. It'll probably get bigger, but it's less than 4 microns, pretty much. And can you guys see that, or is that me projecting? Like, okay, I've seen this before. Like, do you see the spore similarity between this picture,
Yeah, we can see that there's something going on in there, different
and this
shapes.
picture? Yeah.
Yeah, for sure.
All right, cool. But I sent that over to someone that knows Orbilia. I hope to hear back from them.
Okay. Anything else you want to show?
Very inspiring.
No, I mean, I have a bunch of other stuff that isn't quite worked out well enough yet, or that we've already seen a bunch. So
Okay, we'll move on then to.
appreciate it. Thanks, guys.
Yeah, thank you. All right, Paul, you're up.
Okay, thanks, Lila. Let's see. And share there, I think. There we go. Okay. This one I saw the other day. I didn't bring this home. This was on some, these were well attached to this well-rotted hardwood. I did detach one of them there, which doesn't show a whole lot on the back there. You can still see some of the wood remnants on the back. But I was wondering whether this is an early version of the Camarops, the dog nose fungus, possibly, because I've never seen that in an early stage. And I'm just wondering if that's what this might be before the outer layer peels off.
Hmm.
I think it is kind of white beforehand, right? Isn't it?
Yeah, I've never seen the early. I've just seen the sort of the later version after it looks like a dog's nose.
I believe I've seen it and it's white. But
Sort of a
I'm
white
gonna look
haze
into
to it.
that or look at some observations.
You didn't cut one in half, did you?
No, I didn't. I just sort of this was I thought it was just going to be a couple of quick pictures and a sort of a throwaway, but I didn't even bother to bring it home. But anyway, I thought this may be something that someone might recognize.
There are some photos on Michael Quebec of Camarops that look like this.
That do look like this?
Yeah, there's a few photos
Okay.
on Michael Quebec.
Okay, that's where I need to. I tried looking around on the web in general, and I just couldn't seem to find any, young pictures.
A lot of times a Google search will not give you Michael Quebec, probably because it's in French.
Okay. Yeah, I'll have to check that out.
Yeah, I'm looking on INAT at it, and yeah, they do seem to be kind of these white, lumpy structures exuding a lot of droplets before they open up.
Yeah. Yeah, I should have brought this one home and done some dissecting, maybe.
Is it somewhere you can go back and look at it in a day?
No, I don't think I can. I sort of stumbled upon it. It was in a pretty large area. I was just walking around. I doubt if I can get back there, but maybe I will. Who knows?
That's cool. It's kind of right in between. I like it.
Yeah, well, I'll have to. Yeah, maybe I'll see if I can find it again. Let's see. Let me show this one real quick, and I think I've got a correct genus on this, which is something new for me. Crinipellis. It's kind of interesting, sort of hairy, sort of top of the cap there, somewhat zone-like,
Beautiful.
particularly this one up there, and this one as well. It's an older one, and underside. It's been well eaten, but I'm wondering, does anybody know Crinipellis?
Yeah, it's what it is. It's Crinipellis. There's a really small species. I forget the name. The most common species is Zonata, I believe. It's probably what this is.
Okay. All right. Good. Okay. Good.
Yeah, so there's like little hairs along the edge of the cap, I think, with Crinipellis.
Yeah.
Yeah.
This is some pretty low light. I didn't get great resolution on the photos.
You didn't get a lot of resolution, but
Yeah,
it's Crinipellis.
Okay, good. All right. And just one more, maybe. This is one I showed last year. It's orange polypore, which I think used to be Hapalopilus croceus. they want to call it Orantoporus croceus. I found this last year in the same general area. Pretty sure this is on oak. At least that's the literature. It just says that it really does only grow on oak. Got a somewhat decent underside shot here. It's much darker orange on the underside and has these angular pores. So I believe that's what this is Orantoporus croceus.
Yeah, that sounds good to me. Not sure I've ever identified this. I think I've misidentified something as this once, because there's a hypomyces that grows on polypores, that turns the polypore into something that looks like this. And unfortunately, the tests for well, it used to be Hapalopilus, right? You just said the new genus. I forget what it is, but KOH on the Hapalopilus croceus, or whatever the new genus is, goes purple or reddish purple, but it's the same reaction on the hypomyces, which was why I had mistakenly posted on Mushroom Observer under the old name Hapalopilus, but it's this hypomyces. But that's probably what this is.
Yeah, this one stains more red, and the other one is more purple.
Okay, yeah, I think there is a little bit of a difference.
Yeah,
the KOH reaction.
Pretty profound. Is the other Hapalopilus still in Hapalopilus, or has that also changed genuses?
That's what I was wondering the same thing. The Hapalopilus nigilans.
Yeah.
Yeah, it's not nigilans anymore though. It's
Rudolens.
Rudolens,
yeah.
Oh,
But that's
it's
still Hapalopilus.
This one, by the way, doesn't do anything in the dye pot.
I was going to
Oh,
ask
really?
whether this was a
Oh.
dyer.
No.
Oh, that's
surprising.
I've had it years ago from a couple of different places. Found it myself in Pennsylvania in Michaud Forest. Does nothing. Die dud, as Arlene would say.
That's interesting because it does have the KOH color reaction.
Yeah.
Or yes, or ammonia. Same thing. As I remember, it was kind of more of a cherry red in its description, but that's a long, long time ago.
Yeah, I'd say like crimson, but definitely red.
And
Okay,
can you zoom in on those pores again?
The hypomyces goes purple. With the thing that I think is similar to this, that one goes purple with the KOH.
And see, oh gosh, who was it? Karen and Nancy, right? When we were looking at the polypore that you shared, we can see that these pores are a little bit angled, right?
Yeah. Yeah. So these are the kinds of photos we need to be getting.
Yep.
Yeah.
The more information you can provide. I mean, there's a lot of data points, and the more photos of different angles, it's just more data points.
Yeah. Okay.
And also the texture, this looks quite soft, but it might not be as soft as it looks. But that would be also helpful, even though it is young, looks young on the growing edge anyway.
Yeah. The other thing you might find mildly interesting is where I found this. I found this in Druid Hill Park here in Baltimore City. Druid Hill Park is a place that Mary Banning, the self-taught and nearly forgotten mycologist of the 1870s and 1880s, she used to walk around Druid Hill Park and collect things and
that's very
and
cool.
That talk from, I forget who gave the talk about Mary Banning. It was so good.
How do you pronounce it, Lila? From the New York State Museum, which, by the way, will have Mary Banning's drawings on display, her watercolor paintings, through, I think, January of 2026. If you're anywhere near the New York State Museum, you definitely want to go see that. It's free. to get in there.
Patty Cation.
Yeah, I would really like to get up there at some point that's in Albany, you say?
Yes,
In
yes.
Albany, yeah. Yeah.
Cashian, is that how you
Yeah,
pronounce it?
Cation or Cation. Yeah,
Okay.
Kaishian.
Yeah. Yeah, she's gotten very excited about presenting her work.
Mhm.
That's it for me.
Great. All right. Thanks, Paul. All right. Before we go to the email, anyone who joined late, does anyone have something they would like to show from their desktop? William, Doug, Armas? Anybody? All right. Let me get to the email here. All right, Michelle, are you still there? You're up. All
I
right.
am here.
All right, let me share.
And I just joined the group last week, so very, very new. And you guys have probably seen this mushroom frequently, but I just wanted, it's my first one that I ever picked, so I just wanted to confirm what I think it is, which is a chanterelle, but
Yay, Michelle.
yeah. And this was found up in New Hampshire this past weekend.
It's a
So,
beautiful.
and there's quite a few you can see on the ground there. So I only picked one, but yeah.
Wow.
Yep. Chanterelle.
Yeah,
Okay.
yeah. They're coming up here in northern New York, so yeah, it doesn't surprise me. We've had similar weather patterns. The trick is to get them before the bugs if you want to eat them.
Okay.
Yeah,
Best
Good to know.
a little
one of the
worm.
best to edibles.
Even if there's some bug damage, sometimes you can trim away the damaged parts and still end up with, you know, a reasonably good percentage of material out of what you collect, because you can get a lot of them when when they're fruiting.
Mm-hmm.
Cut one in half and top to
Yes.
bottom.
Top to bottom, and then you can sometimes there's insect tunnels that go right through the stalk up through the cap. A lot of times you can just whittle that part out.
Okay. Good to know. I'll keep an eye out in New Jersey for these now that I know.
On the West Coast, the bugs don't really attack the chanterelles like they do east of the Mississippi. So if you're out there, you won't have to.
That's what David Aurora says. But if you're looking for chanterelles in New Jersey, you might find a species that's equally good to eat, but it looks a little bit different than these in that it's not going to have the well-formed ridges on the underside. The so-called smooth chanterelle, which I think now is in northern parts of eastern North America, is Cantharellus flavolataridius, has a more or less smooth underside. And if it stays in situ for long enough and there's a lot of damp, rainy weather and it continues to grow, it'll develop veins, like these thick, undulating veins, unlike these rather straight ridges that are coming down the stalk. So there's a little bit of a difference. So that'll throw you off a little bit if you find chanterelles in New Jersey. It's not unlikely it would be this other species.
Yeah, those are good.
Where in New Jersey did you find these?
No, this is New Hampshire.
Mm-hmm.
Oh, okay.
Yeah, this is the kind I usually find in New England, or at least one of the kinds, because there's more than one species of these more or less classic yellow chanterelles that have the well-developed fertile surface underneath. But I usually, in Vermont, I find one that looks like this, usually under conifers, hemlock or spruce.
And you would
Michelle,
go ahead.
I was just going to say, Michelle, if you're new to this, you'll need to read up on what we mean by mycorrhizal, which means that it's connected to the trees, which means that these will come up in the exact same place year after year, depending on weather and rain and time of year. Very
Okay.
similar time of the month or year too. So that's worth knowing if, you
Okay.
know, in my case, I've got 2 or 3 places and they're all producing now.
Great. Good to know. Thank you. I'll look that up.
Susan, where do you get your chanterelles under conifers in the Adirondacks or mixed woods?
Yeah, I would say probably yes. Yeah, in both cases, I'm thinking there's some white pines and mixed balsam fir, probably some spruce.
And ours predominantly grow with oak,
Right.
where I live.
Yeah,
Yeah.
the smooth chanterelles grow oak hickory woods usually.
So it seems, I forget whether, for the Namaphore in Vermont, whether Rachel Sweeney is coming or whether she's coming to the nymph. If she's coming to one or the other, and if you're there, you want to hear her or ask her about chanterelles. She's been doing a study on them for quite a few years, and she's kind of renamed some of these things.
And we
So-
would really need to DNA test to tell the difference between a lot of these yellow
Right.
chanterelles, right?
And
Those
Rachel
spores aren't
Sweeney
enough.
has done some of that.
Yes, yeah, I know that.
I think in some cases there are some traits you can fall back on, like some of them have white stalks.
And like a greening,
yeah,
like a green halo.
It's a good set of photos. You got the top, you got the side, you got the underneath.
Yeah, we can all agree on this one based on your photos.
Yeah, good job.
Thanks.
All right, well, good. Welcome. Show us more next week.
Thanks.
All right. So we got Dave, Virginia, and Hunter. All right, Dave.
All right, I'm here.
First one up here. Mallocybe, unicolor.
Yeah, this used to be a long time ago. Either this species or something very similar to it was all lumped into a species concept called Inocybe caesarita, I think it was. But now this is no longer a true Inocybe. This has been put into genus Mallocybe. And one of the chief differences between these mushrooms and true Inocybes are the cystidia on the gills. There are these cells on the gills. And here we see, this is the gill edge, and these are the chylocystidia, CHEILOCYSTIDIA. There it is, chylocystidia. So the gill edges are loaded with these chylocystidia, and they are fairly distinctly shaped for the Inocybaceae. A lot of times, the Inocybaceae that are not true Inocybes, they'll just have these like club-shaped chylocystidia. But these are, look at their, like, they call it flexuous, right? Where they're sort of like bendy, subcapitate, where they almost have a head on top, and variously shaped. So that was really, I thought that these were Mallocybe unicolor when I found them. But after I looked under the microscope, I'm really well convinced that's what I have here. It's a close-up of the chylocystidia, and the spores are ellipsoid to. There's some weird word they use, phasioform or something, which I don't usually say because it's easier to say bean-shaped. So some of the spores are almost bean-shaped, which means that they're sort of just like, they're not really symmetric, right? There's like one of the longer sides is a little bit concave. And the spores are fairly large. I think these probably are over 10 microns long, maybe 11 or 12 microns long. I don't think I posted dimensions here because I just didn't bother getting spore dimensions because I'm pretty well convinced I'm right about this being Mallocybe unicolor. So
How did you say the name of that spore shape?
Phasio form or something like that. Bean shaped.
That's fine. Because that is probably like because Fabaceae, is the bean family.
Oh, okay. Yeah, I don't know that. Yeah, so that's where that comes from. I see. Okay. Well, I've learned something else today then. All right, what else do we have here? I don't know what this is, but it might be some kind of Sebacina. It was just this. Sebacina usually grows in these clusters of tightly packed fruit bodies. But this was it. This little thing was growing by itself in some oak dominated woods on the ground. And I laid it down on a microscope slide and didn't get any spore drop. And I think it just found its way into the compost. But if anybody has an idea, I still wouldn't mind having an idea what it is. And, sometimes I get a lead here and I follow up on it and find out the identity of something that I didn't know.
Maybe Telephora.
Yeah, that was, I thought Telephora also. It didn't smell bad.
No, I think he's right in Sebacina.
Yeah.
Yeah.
Maybe Schweinittii.
There's a few different Sebacinas, but Sebacina Schweinittii is really kind of weird. It doesn't always look the same. It manifests in like different shapes for the individual little fruit bodies.
Was this very stiff? Did you say?
Yeah, it was stiff. Yes.
Yeah,
like
that's in line with.
Yeah, like Sebacina is. Yeah, Tilapia is usually a little bit like flimsy.
Not Vialis.
Okay, yeah, not all of them, right? Okay, yeah.
But yeah, I'm not disagreeing.
That one has like weird colors, doesn't it? The Vialis.
Little purple, smells like cat pee.
Yeah, and it smells bad. That's one of the ones I did smell this to test, to see if it might be Tilapia.
Was it more white in real life?
Kind of creamy, beigey, sort of. It's what the photo does justice to the color. That's sort of what it looked like. So it wasn't real white like Sebacina either. That was the other thing that kind of threw me off.
Yeah, it's a little off color.
Susan, you've been trying to hop in here. What were you going to say?
Well, I was going to say, I ran into some things like this a couple of years ago, and there were a lot of variations of what seemed to be Sebacina. The other thing is I keep finding something like this down at the Adirondack Museum, or now the Adirondack Experience, and there's just bunches of them everywhere, and they're never bigger than about 1.5 inches. They're kind of a rubbery hard texture, and they're all in little single individual things. So I've been curious as to what they are for some time. And so anyway, I'd like to know what this is.
Do they ever open up? You know what I mean? Like, do they ever?
No, this is the whole deal right here.
Yeah, just this.
Well, it sounds like you're describing the same thing, Susan.
Yeah, it really does look exactly like that.
Yeah. So you don't know what it is either.
And I assume that's the palm of your hand.
Yeah, that's the palm of my hand. It's small.
Yeah, maybe the ones I get are a little bigger, but still.
The other thing that I was thinking was, I think maybe Susan, you gave me the name, was the Cotylidia.
What was it?
Oh, the Cotylidia thing.
Cotylidia, I think it was.
Something like that, at least. Yeah.
I don't think that was me. That sounds way out of my whatever.
Maybe Dorothy. I think Dorothy may have mentioned.
Yeah, Dorothy sounds smarter than for something like that.
I think she did mention that something Cotylidia.
Yeah. I'm gonna look into that. I don't think that's what it is.
Was that in New Jersey?
I don't think that's what it is either, but just throwing something out there,
Well, it's something for me to look up. At least I'll end up learning something probably, even if I don't figure out what it is.
There is a white one, but this one has like those external tentacles that I don't think would be in Cotylidia. But who knows?
I found something exactly like this. I think I'm going to try to do some work on it this week if I get to it. But Yeah, I threw it into Sebacinia.
I should have
Yeah.
scraped a little and scoped it, but I was busy. There were lots of other mushrooms to look at. There's really a lot of mushrooms. I mean, it's turning out to be quite a good summer of mushrooms.
And just, I'm not saying it is Telephorophyllalis, but I've
Yeah,
seen
for sh-
immature ones next to mature
No,
Telephorophyllalis that
No.
look like this. So I'm not saying that's what it is, but
no, Telephorophyllalis is quite a lot different. But also, yeah, I don't think it's Telephora at all. Well, but a lot of mushrooms are very variable, so
Yeah.
that's part of why it's really good to look at more than one fruiting body if you possibly can. And repetition, same places over years, so you really know the thing, even
Yeah,
if you don't have a name for it.
this was it for this thing. It was growing on the ground near some Cantharellus minor.
Wow, just the one
Just,
fruit body, huh?
just this. Yeah,
Wow. All right,
that's
kind
the
of looks like
weird
three
thing.
fused at the bottom, but
Yeah,
yeah,
because Sebacina usually grows in rather large clusters.
yeah. All right, well,
cool. I mean, everything everyone said, I agree with, but that's a weird one.
Yeah, well, at least I got a few names to think about. Now, this is a really common mushroom. In fact, the common name of this mushroom is the common Satharella. Even though it's no longer in genus Satharella, it's now Candylomyces candylianus. And they grow on my property in the same spot. Like they start in the spring and you get them. But that's the same mushroom. That's why I posted this. Look at this. That's a day after, 20 hours later from the previous photo.
After rain.
Same exact mushroom. Look at it split and it turned up on the sort of the rays and the split turned up on themselves. You see the gills like wrapped around the top. It's just a cool photo. So
Dave,
that's why I shared this one.
Was
just
this
because
after
it's cool.
rain between those two
There
photos?
probably was some rain. I forget. I mean,
That's
it
cool.
rains almost all the time anymore. Actually, though, we just had. Oh wait, no, wait a second. What's the date on this? This
third.
is the
The third.
There may have been some rain on this. I think there was some rain on the second. Yeah, because the weekend, the third and the fourth, were there was no rain. And the fifth also, I believe there was no, but that doesn't count here because that's after this. But I think there was some rain on the second, so that it probably rained and this thing just blew out. It just looks kind of cool though.
It is. Interesting photo. All right.
And was that thing pretty good size? So I
And
find
a moderately sized one. Once it was expanded like that, maybe 2 inches.
Right.
So
Because
this is also on my property. There's a story behind this mushroom. When I first moved in here in 2001, I mowed paths up on the overgrown farm hill that's part of our 4-acre property. And after I mowed the paths, then I would mow. I take the mower up there and mow occasionally to keep the path open. Well, a little oak tree started growing. It was about maybe 10 inches high, and it was right where I was walking behind the mower. It's a walk behind. You just squeeze the handle and it goes, and you go for a walk. And I remember distinctly in just remembering every time I mowed this path, I shouldn't mow that little oak, because oaks are really good trees. And that oak is now probably about 25 feet high. The trunk is maybe getting close to 10, 12 inches wide. And these amanitas are one mycorrhizal species amongst a few that are apparently associated with this somewhat isolated young oak tree. So I've had these sequenced by Fundus, I believe. So that was Sanger sequencing. And this is Amanita crenulata, which is kind of interesting because Amanita crenulata is typically thought of as a fall species, and the colors are usually a little more muted. And sometimes the ones I've been finding up in my spot on my property, and there's another spot as well. There's another spot with a fairly young oak where I've been finding these. Sometimes they are really vividly yellow, and sometimes the stalks are kind of long too. And in contrast to what you expect from Amanita crenulata, which is sort of a short, stout mushroom with sort of a muted color on the cap. They call them the champagne poison Amanita because the colors of both the cap and the universal veil are sort of a champagne color. But this is Amanita crenulata, grows on my property, and I was able to more or less observe the entire coming about of this species being in this spot, because I watched that oak tree grow over the course of 24 years, or well, maybe 22. It may have been like 2003 or so, or four, that little oak first appeared.
Nice.
Yeah, so it's no, the mushroom itself is not totally exciting. I didn't even harvest it. Maybe I should have, just so you can see it. But Amanita crenulata is a very common species. If you look it up online, you're going to find lots and lots of photos. On Mushroom Observer, I probably have at least a dozen Amanita crenulata observations.
Was that your fourth one?
I think there's one more. I'm not sure.
Yeah.
Well, let's see if it's him. Oh, here we are. Yeah. Stecturinum ocratium. This was found on my property also. It's just on a stick. I don't know what kind of stick. Some kind of hardwood, because there's really no conifers where this was found. Now, the best thing about this observation, I think, is going to be maybe this photo right here. This is the hymenium. These are true spines, as opposed to tubes on a polypore that become lacerated and tooth-like you would see in a species called Urpex lacteus, which is otherwise kind of similar to this thing.
Or Hydnoporia olivaceum, which
that one I don't know if I've ever even identified. I need to try to look for that. But these are true spines.
Yeah. Yep.
These are like on a hedgehog. We would say that this fertile surface is hydnoid. So it's like a hydnum. And just to make sure, I laid this down on a microscope slide, and I got spores. And a lot of times with these resupinate things, you lie them down, and a year later that you might find like 2 spores, with your microscope. But this thing laid down an actual spore print that I was able to see. So I was able to scope the spores. I didn't take a picture. I didn't get a good picture of the spores. They're very small. They're like up to maybe 5.5 microns long, but they are ellipsoid as opposed to being like sausage shaped, which a lot of these resupinate things have like really small, thin spores that are just like little cylinders or sausages. But these were ellipsoid. The shape was ellipsoid, which fits Stecturinum ocratium. So that's why I put this one up here. I just wanted to talk about the fertile surface, which is there's a few other polypore type things that have a fertile surface like this. Climacodon septrendin, blah blah. How do you say? How do you say?
Cantharellus, yeah.
Yeah, that one. Yeah, that one's also got, I believe, a truly hydnoid hymenium.
Yeah,
those things are way more capitate. This thing doesn't. It may reflex a little bit.
Yeah,
but yeah.
Oh, well, those things, yeah, they really look like polypores.
Yeah,
they form these like oval
overlapping brackets. Yeah,
an oval shaped area of overlapping brackets. Yeah, they're pretty distinctive, and this is kind of distinctive. A lot of things look like this at first glance, but when but when you see a close-up of the fertile surface, then you've got a pretty good chance that you're looking at Stectrinum ocratium. Ocratium because it's ochre colored, so which also fits as well.
All right,
Okay, that's mine. Thank you.
nice. All right, thanks, Dave. All right, let's see who else. Virginia.
Okay, these are the bird's nest fungi that grow on the mulch, hardwood mulch right outside along our house, and it's covered with them. There's so many of them. And I figured out it was Crucibulum levi, however it's pronounced. And iNaturalist came up with another Crucibulum simile as a suggestion, but then I noticed that they were all from New Zealand, so I figured it couldn't be that, even though they look pretty similar. So those are the eggs, the peridioles inside of the nests when they open up. And they're pale color like that. And the inside of the nest is smooth and pale. It gets dark when they get old, like the one on the right.
Virginia, is this the kind of pubescent one that we saw, the same one that we saw?
Yes, that other one was from New Jersey, from my sister's yard. And I wasn't sure at first because it was so fuzzy. Okay, so here's one of the peridioles, which I picked out and put under the microscope. And that little button shape there in the middle is what you would see in Crucibulum, and a little bit of the string, thin string. And it's the reddish color is not true. That's the camera effect. They're really pale, just off-white. So that's what I call the Crucibulum levi, or I don't know how it's pronounced.
Are you, is this under a stereoscope?
I forget on if I did that one. I think I did, yeah, and I turned it up to 40.
Yeah, that's
Yeah.
probably a stereoscope because
Yeah, it is.
Yeah.
Yeah, I didn't put those under the compound microscope.
yeah, because these are pretty big to begin with for looking at under a compound microscope. You would, one of these would fill up several screens,
Yeah,
under
right.
under
Yeah,
a
I didn't have to
compound
do that.
microscope.
That's just another picture of a different nest with them. They're sort of on its side. And when they're young, they're very fuzzy like that, and that has a covering, which I guess breaks open after a while, and then you can see the eggs underneath. And they're supposed to be very extremely common. I guess it's one of the most common bird's nest. And I think there's more every year in our mulch bed, and now it seems like they're just everywhere on the mulch.
Yeah, once you notice them, then you start seeing them everywhere.
Yeah, well, didn't used to be quite as many, and I had collected some a number of years ago, and I forget which, if it was the same species or not. I didn't go back and look.
Yeah, they're pretty interesting for those that don't know. The raindrops hit these cups, and it causes these eggs, if you will, to
Yeah, to fly out, and then they stick onto something.
Right. Yeah. So that's how they spread.
Yeah, where they land. Yes.
Yeah, the spores are born on the peridioles, right? And
The spores are actually inside
within.
those
Yeah.
eggs.
inside
actually
inside the
spores.
peridioles. Oh.
Yeah, it's really cool if you can get a shot of like take a really thin section. You can get a good shot of those spores within the peridiole, right?
They're inside the peridioles. Yes. the eggs.
Are these things ascos or basidios? I forget. Do you know?
Sorry, I didn't.
I think they're basidiomycetes.
Okay.
Yeah, I think I saw that. When you go down the tree, they're a taxonomy. I think they start out as basidiomycetes and go from there, which I didn't try to follow and study. I mean, why they're still called that. I mean, why they end up there.
Well, I guess probably the basidia are just like all smashed together inside one of these eggs, sort of like what goes on in a puffball, I suppose.
And just real quick, Nancy and Karen put in the chat, what technology do you use to take such great close-up photos of small mushrooms? The stereo microscope is, I think, what we were looking at there, right?
This one, yeah.
Yes. I wanted to check the bottom to look for that little button because that identifies it as a crucibulum. And has that, you can see there just a trace of that little string.
Yeah. And all the rest of this is part of the peridiol itself, right?
That's one peridiol.
One peridiol, and that's the attachment point.
Where it's attached, correct. And so that would be on the underside when they're sitting in the nest.
Wow.
To Nancy and Karen's question, macro lens on a camera or a phone camera, the macro feature can help. Maybe not get to this level, but also sometimes you could take a picture through a loop as well, a 10x or whatever.
Yeah, you can get a $20 attachment for a smartphone that gives you pretty good close-ups.
Oh, okay, great. Thank you.
Yeah. Yeah.
And that shiny spot is because I put it in a drop of water so it wouldn't fly away. So the light from my little lamp reflected on it, left shiny spots.
All right, well, good. Let's see, you said something else, I think, in
One
here.
more. Yes. Okay.
All right.
I went out one morning and right alongside my sidewalk, between the sidewalk and the side of the house, there was this stinkhorn, very fresh and new. It's shiny, but I didn't have my camera and I had to go somewhere. So I didn't get the picture until later in the morning, and you can see it's all shriveled up. But it was just, this is the first time in my life I actually saw one. As much as I used to walk around in the woods, forever, all my life, I don't think I'd ever seen one just nice. So that had just come out.
They like those wood chip beds for sure.
What's weird is the flies never showed up all the
know.
time that thing was out.
Yeah.
There weren't flies on it, no.
No, they never showed up at all, I don't think.
Yeah, I don't know.
Which, that's weird.
Uh-huh.
Yeah, normally, by now that should be eaten clean.
Was it dry? Was it dry that day? Maybe the flies weren't out that much.
Yeah, I don't know. We've had, I think, I don't remember how much rain we had just before then.
Chips look dry.
That's a bigger picture there, the same thing. And the green thing is a termite bait, which we have all around our house. So it just popped up right there.
Yeah.
Oh, wow.
Coming out of an egg down here, if you.
Yeah, the egg would be in the down in the dirt.
Yeah.
So then it dried up and disappeared.
Yeah.
I didn't collect it.
There, yeah. Yeah, might be smelly if you brought it inside. That's a lot of material there. All right.
So I called it genus Phallus. I don't know the species on those at all.
There's another genus, the one that has elegans.
Mutinus.
Mutinus. Yeah, I think this might be a mutinus. It's got like the thin head.
Yeah, there's Mutinus elegans and Ravenelli.
Yeah, there's two of them. Ravenelli, right? Yeah.
Oh, yeah, it was smooth and shiny, like moist looking. The stalk it was very smooth and shiny when it had first come out.
Oh, okay. Smooth and shiny,
Yeah.
not spongy like.
No, it didn't look spongy.
Mm-hmm.
Like now it looks more spongy because it's shriveled up.
Yeah. Okay.
Not quite as red. I don't
Yeah, if we went and looked in the books, we might be able to get to a species on that then.
Yeah, I think how the brown like cap is like looser on one of them.
Yeah, it kind of hangs free on one of them.
Yeah,
Yeah.
I think the Ravenelli.
What's that called? Is it called gleba on the stinkhorns or?
Yeah, it's not a cap at all.
Yeah.
Yeah, the fertile service is gleba, I think.
Mm-hmm. Okay. All right. Shall we move on?
Yep, that's it.
All right. Thank you. All right. Let's see. Hunter. You got a bunch here. Ooh.
Yeah, another Chalcioporus. This is Chalcioporus rubinellus, I think. It's so pretty. And at first I thought the the pores looked angular. So I was like, is this a Zulilus? Then I'm like, no, it's not. And I kind of narrowed it down to Chalcioporus and then got some help via Igor pointing to rubinellus. But I love how there's that light pink kind of coloring near at the stem though. Yeah, right there. It's so pretty. And this is a pretty small specimen. I do have it, and it was a little peppery, as they all should be, I guess. I'm not positive about that.
Are you in Wisconsin or Illinois?
This was in northern Wisconsin. I spent the holiday there. So yeah, yep, it was up near Lake Superior.
I found one of these today.
You did?
Yeah, northern Adirondacks. Yep.
Wow, cool.
Nearly looks identical.
Oh, cool. That's great.
Same exact color.
Yeah, it's really neat how the overlap. It's really fun to see that. I like that. Yeah, it's really pretty.
So cool. I've never seen this. It's very cool that you both found it.
Yeah, I agree.
Yeah, I was with a group of about 13 people, and it was very impressive to them to see the pink, because it doesn't look like much from the top.
Right.
Turn it over, and it's that beautiful pink color.
Yeah, I was.
Yeah, very
really
blasé top.
Drab on top. How would you describe those pores? Would you call those boletinoid?
Subgenus of bolete, right, Igor?
These would probably be somewhere between zircomoid and boletoid. I'm not quite sure. I don't see these things very often. And I also proposed, within the note, an alternative ID. There's a species called Pseudo-Rubinellus. And
yeah, that's what I called it.
Yeah, since I haven't collected these two and sequenced them, I can now really tell the difference because I don't have enough field experience with these species. So there's a cautionary tale. There are two of them. Rubinellus seems to fade more to yellow, like here. So that's why I propose Rubinellus as opposed to Pseudo-Rubinellus. I'm talking about the cap, not the stem.
Okay, so it could be either.
Could be either. I'm not sure how common or uncommon they are. You know, so, but they tend to be, you know, more of, you know, boreal species up north. You know, I think I've collected one in New Jersey, but I'm not even sure if that's what it is, because I couldn't compare my sequence with anything else in GenBank at the time. So a lot of it is going to depend on the type specimens again. It all just boils down to the type collections. Somebody has to grow a pair and then sequence them.
So this was what. There's really not any red on the cap or the pores, right?
It fades. The cap color is very unstable. As
The
far as I
pores
know,
are pink.
they tend to be pink.
Oh, okay. This does look pink. All right. I'm sorry. I have
Yeah,
to
it
read
was
through
quite
mine.
pink.
Looking again.
was
Yeah, that looks
beautiful.
pink. Okay. Yeah, Rubinellus, Pseudo-Rubinellus.
Yeah,
The
yeah. Both of
Bisset's
them
actually.
have.
Oh, God.
I'm sorry. Both of them have pink pores. That's why, you know, you can distinguish them from other species of Chalciporus, but next to each other, there's got to be other stable morphological traits, so you can tell them apart in the field. I have no idea if they have the same spore sizes, but they're twins.
You know, you would think I would know this because the Bisset's used one of my photos of Pseudo-Rubinellus in one of their field guides.
I
I
see.
think it was Pseudo-Rubinellus, but I forget how to tell the difference between the two species.
Right.
It might be habitat. I'm not sure.
I would have said the Rubinellus might be redder on the top and stay redder. Because I think that's one that comes up under a single hemlock tree at the Adirondack Museum.
I think it's the other way around.
Could be.
Pseudo-Rubinellus tends to stay more red on the cap.
Yeah, I think there's one of them is more red on the cap. That's actually
Yeah,
why I
yeah.
brought that up at first. But yeah, but this is clearly one of those two, or something that's lumped into one or the other species concept.
Now, since they were described as separate species, there's got to be some morphological differences or microscopic difference between the two, because if they don't have them, or there's just a broad range, you know, of spores that, you know, fit within each other, then you have to distinguish them by DNA, and, you know, there's a possibility maybe they're the same species. So you got to do the work.
Yeah,
I'm not
that's a
meaning
good point.
you, but, you know, somebody's got to do the work. You know, I mean,
Yeah, that's a good point.
Chalciporus is a small genus. Come on, people. You know, we got to sequence these things, right?
Well, I'll try my specimen and maybe I'll send it to Stephen Russell or something. See, maybe he'll do something.
That would be great.
Okay.
I have no
We're
idea how much
having a
data.
foray. The club here in Northeast PA, we're having a foray on Saturday in the area where I have found. I forget which one. I think Pseudo-Rubinellus, one of the two. If I see any, I'll certainly collect them and save them.
My experience with this is also if you find it in one place, you're apt to find it sort of in the same time frame in more than one place. In other words, it sort
Like
of
we
comes
did.
when the weather is just right. Well, we're a little far apart, but even so, that could be similar weather patterns or something. You're still right, sweetie.
Yeah, I don't even know if these are mycorrhizal, because, you know, Chalciporus is a very primitive, very early bolete, and some mycologists say that these things could be actually saprobic.
Well, I think they
Remember
believe they're parasitic on other fungi.
Or maybe
Yeah,
these
remember
are
that-
Just one of them.
the Piperatoides might be it was parasitic on the Amanita chrysoglima. Remember I showed that a couple weeks ago?
Yeah, all these early primitive boletes are either parasites or saprobes. But yeah, I mean, it really comes out as an early branch, you know, in the Boletaceae, you know, at the base of the tree. It's been established, by multiple researchers, so it all makes sense. They are dinosaurs.
Very cool.
Very cool to see.
Thanks for your help on that, Igor.
My pleasure. And here we go again. Another Lexinum.
I came into quite a few Laciniums, and I was first, well, I was calling this, what was it calling this?
Varicolor.
Oh yeah, varicolor. And I guess that it is, it's now just in sections, scabra, is that correct?
No, I mean, they are, but you've got to do more work to propose that name with that level of confidence.
Okay.
Lexinum is an exceedingly difficult genus.
Yeah, at least you should section it vertically and see
I know.
what kind of staining.
I
is
know.
inside.
Now, yeah, and this other specimen I have does show some staining without even cutting them. But yeah, I failed on that. And I'm sorry. And yeah.
Yeah,
It's
varicolor.
Scabers, though.
Varicolor looks very similar to Snellii, and they both stain blue and red in the flesh. But according to a researcher from Europe by the name of Noderloss, they can be distinguished from each other based on their habitats. Varicolor actually is a European species, and Snellii is an American species. But we found varicolor here in this country. I have a sequence to prove that. And interestingly, even though morphologically they look very similar to each other, kind of these mottled caps, with lighter areas, they're brown, but they have like white or whitish spots or areas on the cap. Sequence-wise, if you look at their ITS sequences, oh boy, they are so very different. It's unbelievable.
What about the Lacinium cyanobacillus?
Who knows what that is? We got to have a sequence.
Okay, because that's supposed to be a lot of blue staining, and I thought, oh, well, maybe not this one, but this other one I'm going to show. I thought maybe might be it.
Yeah,
But
the other one I thought it was something else, but who knows? Again, like I said, you got to really do a lot of molecular work and start putting these puzzles together.
okay, so and for very new people, the Lacinium have these scabers on the stipe, which you see with those black markings, and so they're called scaber stalks, and many are edible, and they're neat. They're at least easy to identify to Lacinium.
The thing about the scabers is that they are like little scales that always point upwards, not down like on some other mushrooms. Do you understand what I mean? That was explained to me by Roy Watling many years ago.
So would like
see those scales, they sort of point up.
Yeah.
In the way they're laid out on the stem.
Yeah, they're actually tiny amalgamations of minuscule hairs that you need a fair amount of magnification to see this.
Yeah, there are tufts, there are fascicles of hyphal, structures. By the way, I think Watling was the one who described varicolor.
Could very well be. He did a lot of work in Boletus years ago.
Yep. Is he still around?
I think he's now 87.
Wow.
I haven't communicated with him since Christmas, but as far as I know, he's still kicking along, although he's having trouble getting out in the woods, which I'm sure upsets him no end.
Yeah.
Yeah. So yeah, and basically, the core of Lexinum, I'm not going to go into the detail why I say the core.
Can we stay on that photo while you keep going, Igor? Can we stay on that photo?
There are two major sections in the core of Lexinum. The ones with the brown caps that grow exclusively with birch trees, And then there are the other ones that have orange or brick red caps that can grow either with deciduous trees, but including birch, or pines. So this is section scabra, which are exclusively birch associated, and the other one is sectional oxynum, which are the orange or the red capped ones.
Igor, what
So
section is
yeah?
what section is albellum in?
Albellum is not a core of oxynum.
Okay.
Albellum used to be albellum used to be lactinellum, which is now polyphyletic, and it sits, outside of the core lactinum clusters. Unfortunately, Ku merged a lot of things back into lactinum, which I thought was a very bad move. But that's where things are. So I still refer to the two core sections as the lactinum sensu stricto. Everything else for me is lactinum sensu lato.
Okay, yeah, because albellum is actually grows with hardwoods, even though it looks more like something in section scabra.
Correct. So is Longicurvipes.
Yeah.
Which is
Yeah,
another
okay.
oddball.
That's an oddball one. Yeah.
Yeah.
It doesn't feel like it fits in, lactinum proper.
Correct. They're not section scabra or lactinum. They are. If you still accept the idea that lactinum is this very variable genus accommodating very weird things, including some of the secotitoid or things that grow under the ground, truffle-like species from Southeast Asia and Australia and New Zealand. Yeah, you can say that then, lactinum, longicurvipes, and albellum are in lactinum. But I have a strong feeling that eventually, when people are going to start sequencing whole genomes, that genus, as it is defined by Kuo, as at this time, is going to be broken up into other genera, back to what it should be, because collapsing things with DNA seems to be unnatural. DNA is meant to split things, not to put them back together.
What about hemilexinum? Is hemilexinum related to lexinum genetically, or does it sit?
No, hemilexinum is a part of Xerocomoidae, which is a different subfamily.
Okay, all right,
that makes sense.
Mm-hmm.
It's all about this convergent evolution.
Is Hortiboletus also in that kind of family?
Hortiboletus is Boletoidae.
And the only reason I wanted to say zoomed in on this photo was because I was trying to figure out if anything was coming out of the lenticels, whether it was fungal or maybe one of the scale warts kind of working its way out. But now that I'm looking at it, don't really have an opinion.
Yeah, clearly you can see the birch. So birch was around, and this thing only grows with the birch. Unfortunately, there's no staining in the stipe, so we cannot really say exactly what this thing is. It could be neither Snellii or varicolor. It could be something else. Smith and Thiers described a gazillion lactinum species from Michigan, which is probably going to be dwindled to 20% of what they described originally once the DNA smoke clears or the dust settles. But we got to really work on that. I mean, we already have the data. We just have to interpret it correctly. But again, the cautionary tale is when you're in the woods trying to ID your lactinum, I don't think we have enough knowledge to put names in these for the most part.
All right.
Well, we got some more, so.
Yep.
Yep.
Oops, that's the same one. Oh, again, the same one.
Those hinges need some greasing.
Yeah, and I'm sorry for dragging on the lexinum conversation, but the orange capped ones, the ones that I find in the pine barrens, I've never had problems with. But should we use caution when we find the orange capped lexinum type things? Do they give most people GI distress?
No, I think it's I think that idea is probably flawed. The pine ones are fine. The ones that grow with the aspen, I would be more careful about, because again, there were reports many years ago from Colorado, of all places, where there's a lot of aspen, where somebody allegedly got poisoned by these things.
Yeah, I've eaten those insignia or pseudo insignia, whatever they are, out in Colorado and Montana. I didn't have any problem. Neither did Karen. We had no problems with. I think
I did
Lexinum needs to
too.
be
It was
really
good.
well cooked. The stems are quite fibrous, and I think if they're not really well cooked, they might be a little hard to digest.
Right.
I have somebody up here who has trouble with the red cap, the orange Antiocha kind of things.
Sectional Lexinum. Roy Holling, I remember, told us by me, I mean a bunch of people back in the day, maybe it was at the Comofore, a story, because he, along with somebody else, described a very interesting species of Lexinum. Interesting because of where it was and what it was growing with, but those were just looked identical to what we find here in the Pine Barrens in terms of the overall morphology. He found it in Costa Rica. It's called Monticola, Lexinum monticola, and there were tons of them there growing in the mountains with some shrubs, not even trees. And they cooked them and they ate them, and his co-author was fine, but Roy Holling wasn't. So, whether it was poisoning or just like a idiosyncratic reaction, the jury's still out. But again, the message is what's edible for me is not necessarily edible for you.
Yeah.
Yeah, Lexinum monticola, a very attractive species. Look it up.
Cool. Sorry to digress, but yeah.
No, there's actually more. I've got pictures of a red cap too in the group, but not this one. I mean, like a red cap Lexinum, but so we're gonna see that.
Yeah.
too.
So now we're getting in the realm of these yellow and red boletes with ballooning pores or whatnot.
And so again, with a suggestion from Igor, I'm thinking this is Boletus pseudochrysin toroides.
Right.
It's a blue stainer. It's bigger than the use those little Boletuses that I find. And that's it. And I don't have the proper cross sections, and I didn't collect the specimen. And these are Illinois.
Yeah, that's too bad because I was hoping you would have a sample.
Oh, sorry.
Yeah, is the book closed on this not being Hortiboletus?
Well, here's the thing. There's one sequence collection from Missouri, and I sequenced it. I mean, it wasn't me, but I submitted for sequencing to somebody. And that came back as very distinct, and there are no matches still in GenBank for either LSU or TEF1 genes. And I identify them as Boletus pseudochrysantoroides. Now, we do have the genome of that species from Michigan, because the Michigan herbarium has been sequenced. And I was trying to locate the TEF1 sequence within that genome scaffold, and I just couldn't find it. I was pasting in small sections from various parts of TEF1, and I just didn't find it in the document. It's just not there. So I've got to contact the researcher who has the genome and ask him if my sequences match the TEF1 sequence of the holotype of Boletus pseudochrysantoroides, or Boletus, I should say.
Don't Boletus mushrooms have spores with interesting ornamentation on them? I think
Yeah,
it's
they do.
Boletellus.
They do mostly. They tend to be ridged or ribbed.
Yeah, ribbed. Yes, yeah, like these longitudinal ribs.
From end to end.
Yeah, end to end. Yes. Mm-hmm.
Yeah, but this one is way too red. It's bigger, and overall, it just doesn't quite look to me like a Hortiboletus.
You saying too red to be Hortiboletus? Because I mean, some of the ones I find are pretty red.
Well, you can click on the name below, and it's going to give you all the collections of If then click on this name in the
Yeah.
about this taxon.
Oh,
where
this is
you-
on Mushroom. This is on Mushroom Observer. Oh, good. Okay.
Right, yeah. Click on this thing,
Yeah.
right? And then scroll to the right first. Oh, either this one, yeah, the one that's in the second row in the left. This one, yeah. Click on this thing, and that's the one that was sequenced.
Oh, okay. Nice.
So as you can see on the
That's
right, I
pretty
have
similar.
LSU and TEF1 sequences. They're both in GenBank. And about maybe 2 or 3 hours, I blasted again the TEF1 sequence, and there's nothing closer than 92%.
Well, and this does look like Hunter's mushroom. It's got the very, very thin, small cracks on the cap, like Hunter's does. And these are kind of big. Yeah, these are bigger than Hortiboletus.
Yeah.
Yeah, but these were quite big that I found too. I was like, ooh, yeah.
Oh, okay.
Well, if you remember where you found them, if you go there again, it would be nice to have another collection.
I will keep my eyes open.
Yep, please do.
Yeah, really colorful, beautiful.
Yeah, it was a pretty one.
All right.
So Trichales rudulans, northern Wisconsin, and just pretty mushroom. And I think
Plums and custard.
yes. So not much more to say really. Are
Yeah,
these
it's a fun name, plums and custard.
Yeah, supposedly it does not taste like plums and custard, though. It's got kind of not a very good taste.
No, it's just an English, common name.
And
Don't
these
eat
are
those.
Probec, right?
Yeah,
Yeah.
they grow on wood.
On wood, but these didn't really appear to be, but
A lot of times you'll find them on the ground from buried wood.
yeah. Yeah, they were really beautiful.
Yeah.
I like finding these. Yeah. Such a pretty mushroom.
Yeah, nice
Beautiful.
natural looking photo there with in situ with the one turned up. Very nice. Like a field guide type photo here.
Yeah, all in one shot. You get the ornamentation on the cap on one in the lower left. You get the coloration on the stipe on the one on the right, and gill attachment, all that gill color.
As nice a photo as this is, to remove that leaf would have helped a lot to see the base of the stem.
Yeah, I was going to say that actually. Yeah, that's the
That was
one
something.
flaw. Yeah.
Groom your debris in your photo, especially if you're going to use it for identification.
Yes. Good point.
But it is a good, it's, a good natural shot, and the color is really good.